It's a real pleasure to introduce the next speaker, Kelly Nguyen. Have I said that correctly? Yes, thank you. So Kelly has been really a true inspiration. She did her PhD in Australia. She did her undergrad in Australia, then she moved to do a PhD here at the LMB with Kiyoshi Nagai, where she worked on the spliceosome. Then she moved to UC Berkeley to work together with Caitlin Collins and Eva Nogales, where she worked on telomerase. She then started her own lab here at the LMB only 3 years ago. And the work of Kelly's team has already provided some very fundamental insights into the structure and function of human telomerase. They solved the structure of human telomerase at a very high resolution and found that histone H2A, H2B dimer is actually a component of the telomerase holoenzyme, which may have implications, important implications for the function of this enzyme. And lately, Kelly's team has actually solved the structure of the complex components of the complex called sheltering, which actually recruit telomerase to chromosome ends. And this task has been deemed impossible by the field until Kelly's team has proved them wrong. So this will be great to hear. Thank you, Kelly.
So thanks, Anna, for the very kind introduction. Can you hear me at the back? So it's my great pleasure to be here and following all these amazing women scientists at the LMB. So to start with, I'm going to tell you a little bit about my journey and my time in Kiyoshi's lab and how it's been upon my return here. So to get here, I kind of make a very long journey that go kind of across 5 countries and 3 continents. And but I have been very fortunate to get this opportunity. And my scientific career started with my time working with Gottfried Alting at the ANU in Australia, where I was transitioning from being a chemist to a biologist. So I spent 2 years in Gottfried's lab learning biology in his lab. And then fortunately during this time in 2008, he invited Tom Stein to come and give a talk. So in Australia, we don't often have speakers internationally to come and give a talk. So that was one of the life-changing experience for me. It was so amazing that, I decided I want to do something similar like this. So then I very naively kind of applied to the LMB and was got an opportunity to work with Kiyoshi Nagai, who, as you know, sadly passed away. And Kiyoshi's lab worked on the symposium, and the project was extremely difficult. So none of us finished the PhD within the 4 years. So we all got an extension, myself included. So then after that, I moved to Berkeley to work with these 2 amazing scientists on human telomerase. You hear more about that later. And then towards the end of 2019, I returned to the LMB to have my lab, and this is our lab photo as of 2 weeks ago. So then my time at the LMB is divided into 2 phases, and it has been quite different in these 2 phases. So when I started in Kiyoshi's lab, I was completely naive. So I started, this is the photo of my first day in the lab. We went out for dinner. And a month or so after this, I spilled the whole bottle of big carb ethanol, and the whole floor smelled for weeks. So I was known as the new student of Kiyoshi who spilled the beer me. That was great. But I had to say that at the LMB, I've learned so much through these 6 years, and it really takes a village to raise a PhD student like myself. And that involved many, many people that I cannot show the pictures here. But this is Kiyoshi kindly shares his office with me, 308, and many other people. We have time in Frankly as well, that didn't show here, but this is in Frankly and many of the last events. And after that incident, I often ask Kiyoshi, is there any hope for me? And the people in the lab often say that there's still hope for you. So that was very reassuring. And I learned also from this time it's never too late to do something new, and also to take a risk. And Kiyoshi is really, his lab was working on a very challenging project at the time. And they really embraced it. Everyone struggled, but everyone enjoyed it very much. And he took great pride in his science and take great responsibility what we put out there, as well as they offer a lot of kindness, generosity, and humility. So that really defined my first phase at the LMB. And even with that hopeless beginning, I was able to solve a number of structures in his lab, and that's also thanks to the guidance of some of the people I show down here. And just to know that this middle structure here is really beginning of the revolution in Nguyen in the spices field. But at the time, I thought I'd done enough, and I wanted to try a new challenge. So I went to UC Berkeley to work with Kathy Collins and Evan Nogales. And then that project then took me back to the LMB to start my own lab. So when I started my lab, I imagined I had a very happy time, and this is one of the celebration of our paper. But this is how it ended up. So 6 months into starting the lab, we all go into lockdown, and this is kind of we couldn't have lab pictures, right? So this is a nice one. Sorry, Samray, we should put a mask on you, but yeah. So then so kind of quickly touch on some of the challenges of starting a lab in the pandemic, because many of my colleagues, as well as friends at the LMB that went on to start a lab have, and this can be point of discussions in the panel discussions. So first is like you had to supervise everyone with no direct communication or mentoring, and there's no discussions with colleagues that I was really looking forward to. And recruitment is already hard, but it's even harder. And orders, sometimes we forgot about it because they take months or years to come. And also, I find myself saying, in the old days, we used to do this and that. And I realized that most of my lab members have not had the full LMB experience. And I really hope that we can restart some of these cultural things that we do at the LMB, and now things are back to normal. And everyone is affected in different ways. So you have to manage 5 different situations at the same time. But again, my lab has been very lucky. The science continues under the circumstances. And I'm going to tell you a little bit about our work on telomere biology and exploring the ends of the chromosomes. And as I said, I started this work with these 2 amazing scientists. And the work that I'm telling you is done by this great group of people who bravely joined me through the pandemic. So what do we do? So we study this region, which is located at the ends of chromosomes, called telomeres. And they're extremely important for protecting the genomic information. But what it turns out that they quite unique in a sense that they consist of repeats. In humans, this is GTTAG. This can range between 2 kilobases to 100 bases in mammalian cells. However, even though they're very important for protecting the chromosomes, they're extremely problematic in 2 ways, 2 main ways. The first is that they resemble DNA breaks. The cell had to be able to tell this is DNA break or chromosome ends. The second is they lead to incomplete genome replication. So that brings me to the 2 problems my lab is trying to address, which is how do cells distinguish the ends in DNA breaks? And the second is how are the ends replicated? And for today's, we're going to talk more about the second question. So as kind of, Cynthia's talk kind of leading really nicely towards telomeres and telomerase, because what we know from many years of work is that telomere length is kind of a molecular aging clock. So when the cells are young, we have plenty of telomeres, but over time they get shortened because of the incomplete genome replication. When the telomeres become about 2 kilobases or so, this will lead to all these chromosome fusions, and the cells can no longer live very long. So it leads to cross arrest and genome instability and cell death. And unfortunately, this is pretty much happening for all somatic cells, and that's most of what's in our body. But the question is, how do telomere shortening overcome? And you might remember from Cynthia's talk, it is overcome by a molecule called telomerase, which is discovered by my scientific grandmother and great-grandmother, Liz Blackburn and Carol Ryder. they showed that telomerase turn back the cellular aging clock. And this is in humans, unfortunately, is only limited to cancer stem cells and germline cells. So what these cells have in common is that they activate telomerase, which adds a telomere back to the chromosomes. And in these cells, they have stable telomere length over a very long period of time. And therefore, this is often, kind of dubbed as the immortality enzyme. So you can already see that there's multiple ways you can think about telomerase as a therapeutic target. So first is that it's mostly activating cancer cells. You can very specifically target telomerase to cure cancers. On the other hand, as Cynthia has told you, if you upregulate telomerase in normal cells, you can prolong the cell age. And also another point that we know that there are many mutations that lead to premature aging diseases are affecting telomerase. So, but currently as we speak, there's no drug on the market that target telomerase. And one of the major issues with studying this enzyme is that up to very recently, we don't even know the full picture of what's in this enzyme. So this is kind of the task that I wanted to go to Berkeley to address. And over the years, there's been a number of milestones, and I'm going to highlight a few here. So human telomerase was discovered in 1989, and thus has been shown to consist of these two components, telomerase RNA and the reverse transcriptase component. And it took until 2013 when Daniela Rose and Joachim Linger's lab saw this next-gen structures of human telomerase to 30 angstrom. So when I was in Kathy and Eva's lab, I was able to take this and refine it to 8 angstrom. For the first time, we can define most of the composition of this complex and where all the parts are. And then in my lab at the LMB, through the lockdown 2020, we now refine this to a level that we can see atomic details. And really, this is kind of one of the things that was amazing is that after 3 decades, we still discover new subunits. And in this case, I want to highlight this, this histone that we found here in the structure. So in the March 2020, we went into lockdown, and then we kind of had, got the grid collected by EM facilities, and were able to solve the structure and found this unmodeled density that's kind of sticking out here. And then we found that this is actually histone H2A, H2B dimer. And that gives us a very surprising view of human telomerase. This is right in the catalytic core of human telomerase next to the reverse transcriptase subunit. And what we found is for the very first example of histone binding to RNA, you can see here on the right. Currently, we're working towards understanding what it's actually doing. And the next part that we kind of also want to understand following this work is how it gets recruited to telomeres, because most of the time telomerase is not at the telomeres. So this process is promoted by 2 factors at the telomeres called TP1 part 1. So they recruit the telomerase in an ATM dependent manner at the end of the S phase. And to understand this process, a very talented graduate student, also a postdoc in my lab, they team up and solve the structure of human telomerase in complex with these factors and showing how these interact with telomerase and bring it to the telomeres. And so this is also, I want to also highlight that, Shraddha, who's the new in Visual Aid, who's now leading the Visual Aid team, also done a very nice illustration showing how TP1 part 1 brings telomerase to the telomeres. So yeah, if you haven't met her yet, I recommend you come and talk to her as well. So there's still more things to come, but for today's talk, I just gonna highlight a couple that we have been able to do through the pandemic by this very talented group of people in my lab that here now that's how it's going. Very, very happy to have this group of people, and everyone has been so resilient and remarkable. And I would like to also thank a lot of people from all the labs that have been in, as well as many at the LMB that I cannot name all of this. And I also want to kind of pay tribute to our friend, Chris Ulbrich, and also my mentor, Yoshina Guy, who sadly passed away the last 2 years. And And also another highlight recently post-pandemic is my parents were able to come for the first time. And yeah, this is a mandatory picture for them before they go back to Vietnam. And I also wearing this pin that's being passed down by Laurie Passmore and Rebecca Voorhees. So this is the Sturbridge Award, which is passed on for women scientists. So yeah, thank you very much for your attention.
Thank you very much, Kelly. So questions?
Hey, Kelly, thank you for a great talk. I was just wondering if you had any idea whether histone modifications might change the interaction with telomerase.
Yeah, so the kind of unfortunate thing is we only see the core histone part. So we don't see any tail, so then we cannot. And when we try to do mass spec, we have all kinds of peptides of variants. So yeah, but that's something hopefully with some in vivo work we're starting now, we were able to find some.
More questions? Yeah, sure.
Hi, thanks for the nice talk, Kelly. My question is very basic, maybe. You said there was a missing density. So how did you decide, like, how did you come across that it could be histones when it was not known?
Yeah, so that didn't explain this very much. So thanks also at the LMB. So when Alan Brown in Venki's lab solved the ribosome structures, the way they did it, they took this map and then they searched over the BDB and see if anything that's known fit in there. I did exactly the same, so completely unbiased.